Hello students, I hope you all are fine and happy. We are reading chapter number 2, Is Matter Around Us Pure? We are revising the different separation techniques that are used to separate the different kinds of mixture. Now the technique that we use to separate a particular kind of mixture, it depends upon the properties of the elements or the substances that are present in that mixture. In our previous video, we have read how can we separate a mixture of ammonium chloride and salt. So we know that ammonium chloride is a substance that undergoes sublimation. Sublimation means it directly changes into a gas. Whenever any solid changes directly into a gas, that is known as sublimation. So ammonium chloride is a sublimable substance, means it can undergo sublimation. So for separating the ammonium chloride and salt, we use the sublimation method. So we keep the mixture in a china dish and then we cover it with an inverted funnel and we close the hole of the funnel by inserting a cotton plug and then we start heating it. So the ammonium chloride gets converted into the ammonium chloride vapors and those vapors get condensed on the cool walls of the funnel. Then we take out the ammonium chloride from the wall. Now the salt, being a solid and having a very high melting point and boiling point, it does not get converted into vapors, so it remains as a solid in the china dish. Now the next topic that we read is how can we separate the different dyes that are present in the black ink. So we know that ink is a mixture of different dyes that are dissolved in water. Now the different dyes that are dissolved in water, they can be separated by the method of chromatography. So in the chromatography, what we do, we take a chromatography paper and then we put a spot or we make a spot of the ink on the paper and then we hang this paper in a glass jar that is containing some water. Now slowly what happens, the water starts rising up in the chromatography paper, and when the water rises up, the different inks that are having different solubilities, they also rises up in the chromatography paper and they get separated. So for separating the different dyes from the ink, we use the chromatography paper. Now it is a very important method of separation. Now the chromatography is also used in the forensic laboratories to test the poisonous substances that may be present in very small amounts in someone's stomach. And it is also used in testing the COVID. Like there is a rapid test kit that uses the chromatography. The next topic that we read is how can we separate a mixture of two miscible liquids. Now miscible liquids means those liquids which gets mixed with each other properly and they don't have a layer of separation or a boundary of separation between them. Now the two miscible liquids, they can be separated by using the distillation method. So what we do, we pour the two miscible liquids in the distillation flask and then we add a thermometer and then we heat the mixture. So what happens, though both the when we heat the mixture, so the different liquids that are present, the different miscible liquids, they are having the different boiling points. So when they have different boiling points, so they will boil or they will convert into vapors at different temperatures. So the liquid that will have lower boiling point, it will boil first and convert into vapors. So those vapors we will collect in a beaker by condensing those vapors. How we will condense? We will add a condenser in which the cold water will keep on flowing. So that cold water will cool down the vapors of the miscible liquids, and then those vapors will get condensed and convert into a liquid which will be collected in a beaker. Now the another liquid that will remain in the distillation flask. Now today we will learn how can we obtain pure copper sulfate from an impure sample. Today we are going to learn how can we separate pure copper sulfate Now in short, we write copper sulfate as CuSO4 from an impure sample. So the method that is used to obtain pure copper sulfate from an impure sample, the method is crystallization. Name of the method is crystallization. Now in this method, the crystals of the solids are formed. So let's learn about the method. Now what we do, first of all, we will take around 5 grams of impure copper sulfate. We take 5 grams of impure copper sulfate. Then what we are going to do, we will take it in a china dish. We will take this 5 gram of copper sulfate in a china dish. Then we will dissolve it in minimum amount of water. Then we will dissolve it minimum amount of water Now there may be some insoluble impurities. That are present in the impure sample of the copper sulfate. Now the insoluble impurities, they will get separated. How we will separate them? We will filter this. We will filter this solution. We will filter it. So the large size insoluble impurities, they will get filtered. Now the solution, what we will do? We will take it again on china dish. We will take it on a china dish again. Now what we will do? We will heat it. We will start heating this mixture of copper sulfate. We will start heating it. Now when we will heat it, what will happen? The water that will be present in this mixture, it will start evaporating. The water will evaporate. Okay. Now when the water will get evaporated, so slowly the solution will get saturated. It will get saturated. You obtain a saturated solution. Now how we will check that the solution has got saturated or not? What we will do? We will dip a glass rod. We will take a glass rod and we will dip it in the mixture. We will dip a glass rod. Now if some crystals will be formed on the glass rod, it means the solution has got saturated. Then when the solution will become saturated, at that time we will stop heating it. We have to check it again and again until it will get saturated. As soon as it will get saturated, we will stop heating it. Now we will allow the mixture to cool down for a few hours. Approximately 1 day we will keep it for cooling. So what we will observe? We will observe that the crystals will be formed. At the bottom, the crystals of copper sulfate will be formed. Now this is the pure copper sulfate. These crystals will be of pure copper sulfate. So in this way we can obtain the pure copper sulfate from an impure sample. What we have to do, first of all we have to take the impure copper sulfate and then we have to dissolve it in minimum amount of water, and then we have to filter it to remove the insoluble impurities, and after removing the insoluble impurities we will keep it on a china dish and then we will heat it. When we will heat it, the water will get evaporated. So after time and time again, we have to check for the saturated solution. How we will check? We will dip a glass rod time after time, and as soon as few crystals will be formed on the glass rod, it means the solution became saturated. Then we will stop heating it. When we will stop heating it, then we have to allow it to cool for few hours. Then we will observe that some crystals will be formed, and those crystals will be of pure copper sulfate. Now this process is known as crystallization, means forming the crystals of pure solid. So we can obtain a pure solid from an impure sample by the method of crystallization. Like the pure salt, we obtain the pure salt from the seawater, by the method of crystallization. Similarly, if we are having an impure sample of sugar, if we are having impure sample of sugar, so we can purify it, we can obtain the pure sugar by the method of crystallization. Now we can obtain the pure salt from seawater by evaporation also, but what will happen? There will be many impurities in that salt. If we do it same with the sugar, then also some impurities may be left. And when we will evaporate the water to obtain the sugar, so to make it dry, we have to heat it to high temperature. Then the sugar may get decomposed. There are some solids which get decomposed when they are heated to a very high temperature. So this method of crystallization is best for obtaining the solids from their impure sample. Now we will discuss some questions that in that we have to tell the separation technique that we will apply for the separation of the mixtures. So the first mixture is sodium chloride from its solution in water. So sodium chloride, do you remember what is sodium chloride? Yes, it is salt that we use in our kitchen, or also known as the common salt. So if we have to separate the sodium chloride from its solution in water, then we can either evaporate the water or we can use the crystallization method. Okay, first of all we will filter it. Because if there will be any insoluble impurities, so that will get separated by the filter paper. Then after that we can evaporate the water, or if we want, we can use the method of crystallization. So do you remember how we will use the crystallization method? So first of all, we will heat it. We are going to heat this mixture to make it a saturated solution. Now we have to check it time and again whether the solution has become saturated or not. Now for the saturated solution, what we will do, we will dip a glass rod in the solution time after again. So when the crystals will be formed on the glass rod, so it means the solution has become saturated. Then we will stop heating it. So after that we will allow the mixture to cool down for some time. Now when the mixture will get cooled, so slowly we will observe that the crystals of the sodium chloride, that is common salt, they will be formed. So in this way, either by the evaporation or by the crystallization, we can get the sodium chloride from its solution in the water. Now the next is we have to separate the ammonium chloride from a mixture that is containing ammonium chloride and sodium chloride. Okay, so here the components of the mixture are ammonium chloride and basically the common salt. Now we know that ammonium chloride is a sublimable substance, means it undergoes sublimation. So we will take both these substances in a china dish, and then we will keep that china dish over the tripod stand, and we will keep an inverted funnel over the china dish, and we will close the pore in the funnel by using a cotton plug. Now we will start heating the mixture from the bottom. So when the mixture will be heated, so the ammonium chloride, that is a sublimable substance, it will change into the vapor forms without converting into a liquid, and then it will convert into ammonium chloride vapors, and then vapors being lighter in weight, they will rise up, and when they will come in the contact of cold walls of the glass funnel, so they will get deposited on the glass funnel in the form of solid ammonium chloride, and the sodium chloride will remain in the china dish in the form of a solid. So in this way, by using the method of So in this way, we can separate the ammonium chloride from the sodium chloride by using the method of sublimation. Now the next part is we have to separate the small pieces of metal in the engine oil of car. Now the small pieces of metal, they are insoluble impurities, and we can separate them by using a filter paper, or we can say we can separate them by the method of filtration. Okay, because the metal pieces are large in size, so they will not pass through the filter paper, whereas the engine oil will pass through the filter paper. So the metal pieces will remain on the filter paper. Now the next part is we have to separate the different pigments from an extract of flower petals. Extract of flower petals means the liquid that is taken out from the petals of the flower. So we have to separate the different pigments that are present in it. Now we can separate the different pigments from the extract of flower petals by the method of chromatography. We'll use here the chromatography. What we will do, we will make a spot of the extract of flower petals on the chromatography paper, and then we are going to suspend this chromatography paper in a glass jar which is containing some amount of water. So as soon as the water will rise up in the filter paper or the chromatography paper, so the different pigments will also start rising up with the water. Now the different pigments will have the different solubilities in the water, so they will get separated by the chromatography method. Now the next is butter from curd. Now the butter from curd can be separated by the method of centrifugation. What we can do, we can put the curd in the centrifuge tube, and then we will switch on the centrifuge machine. So what will happen, the machine will rotate the curd at a very fast speed. So butter being heavier, it will get separated from the curd. So the method is centrifugation. Now the next is oil from water. So do you remember what type of mixture is oil and water? Yes, they forms are immiscible liquids. They are both are immiscible liquids, so their mixture is immiscible. Now we can easily separate them by using a separating funnel. What we will do, we will take a separating funnel and we will fix it on a stand. Then we are going to pour the mixture of oil and water in the separating funnel. Now we will allow the mixture to stand for some time. We will not disturb it for some time. So what will happen? The oil and water, they will form the different layers. Now the water being heavier, it will be present at the bottom, whereas the oil being lighter in weight, it will keep floating on the water. Now the bottom layer of water, we will collect it by opening the tap that is in the separating funnel. So as soon as the oil will reach the tap of the separating funnel, so we will close it. Now we will collect the oil in a separate container. So in this way, by using the separating funnel, we can easily separate the oil from water. Whenever we are having a mixture of two or more immiscible liquids, we can easily separate it by using the separating funnel. Now the next is, how can we separate the tea leaves from tea? Now the tea leaves being solid and the tea being a liquid, we can separate them either by using a filter paper or we can use a sieve. So we can either use a strainer or a filter paper to separate the tea leaves from the tea. Now the next is iron pins from the sand. Now when we have to separate the iron pins from the sand, then what we will do, we will take a piece of magnet. Why we will take a magnet? Because the iron pins, they are a magnetic substance, so they will be easily attracted by the magnet and they will get separated from the sand. So what we will do, we will take a piece of magnet and we will bring it close to the mixture. So iron pins being a magnetic material, they will get attracted by the magnet, whereas the sand, which is not a magnetic material, it will remain there. So these are the different methods which we will apply for separating these mixtures. Now the next question is, Pragya tested the solubility of three different substances at different temperatures and collected the data as given below. Now the results are given in the form of a table as grams of substance that are dissolved in 100 grams of water to form a saturated solution. Means she has taken the substances in grams and she has dissolved it in 100 grams of water for forming a saturated solution. Now the first part of the question is, what mass of potassium nitrate would be needed to produce a saturated solution of potassium nitrate in 50 grams of water at 313 Kelvin? Now we will look in the table that how much potassium nitrate is soluble in 313 Kelvin, or we can say what is the solubility of potassium nitrate at 313 Kelvin. So the solubility of potassium nitrate at 313 Kelvin is 62. So 62 grams of potassium nitrate can be dissolved in 100 grams of water at 313 Kelvin. Now we have to find out how much potassium nitrate would be soluble in 50 grams of water at the 313 Kelvin temperature. So that will be half of the substance that is dissolved in 100 grams of water. So if we will divide 62 from 2, then we will get the answer 31 grams. So 31 grams of potassium nitrate would be needed to produce a saturated solution of potassium nitrate in 50 grams of water at 313 Kelvin. Now the next part is Pragya makes a saturated solution of potassium chloride in water at 353 Kelvin and leaves the solution to cool at room temperature. Now what would she observe as the solution cools? We have to explain it. Now 353 Kelvin. This temperature is more than the room temperature. So when she will prepare the saturated solution of potassium chloride at 353 Kelvin and she will allow the solution to cool to the room temperature. So what will happen? The temperature of the saturated solution will decrease. And we know that when the temperature decreases, the solubility of the substance also decreases. So when the potassium chloride solution will be allowed to cool at room temperature, so its solubility will decrease. So she will observe that some crystals of the potassium nitrate will be formed, or we can say some of the potassium nitrate will get separated and it will be observed as a precipitate at the bottom. Now the next part is find the solubility of each salt at 293 Kelvin. Now which salt has the highest solubility at this temperature? So we can check in the table that the solubility of potassium nitrate at 293 Kelvin is 32 and the solubility of sodium chloride is 36 grams at 293 Kelvin. Now the solubility of potassium chloride is 35 at 293 Kelvin and the solubility of ammonium chloride at 293 Kelvin. It is 37 grams. Now we can see here the salt that has the highest solubility at 293 Kelvin. It is ammonium chloride. Now the next part is what is the effect of change of temperature on the solubility of a salt. Now if the temperature of the solution increases, then the solubility of the salt increases, means more salt can be dissolved in that particular solution. And if the temperature of the solution decreases, then the solubility also decreases, means some of the salt will get separated from the solution in the form of the crystals, or we can say in the form of the precipitate. Okay, so as the temperature increases, the solubility of the salts increases, and with the decrease in the temperature, the solubility of the salt also decreases. So that's it, students. Now our chapter 2 is over. In the next video we will start the revision of the chapter 3. Thanks for watching. Bye bye. Take care.